Let us first examine about the inheritance of comb pattern in poultry. Some of the first evidence that a trait can be influenced by more than one gene was obtained by Bateson and Punnett from breeding experiments with chickens. They clearly demonstrated that two independently assorting genes can control a single trait. That means different combinations of alleles from the two genes result in a different phenotype. Bateson and Punnett analyzed the three comb pattern of chicken known to exist at that time. Chickens have a variety of comb types. They are rose, walnut, pea and single. Rose comb found in Wyandotte breed, pea comb in Brahma breed, single comb in Leghorn breed and walnut comb found in Malay breed of fowls. Rose comb is a classical monogenic trait of chickens and varies in different breeds. The rose comb is a fleshy, tube-shaped, solid comb that extends from the beak to the back of the head with a pointed spike. Top surface is slightly convex and with small rounded protuberances. The front portion of the comb is covered with small round bumps terminating in a tapering spike which may turn upward, follow the contour of the head in Wyandotte's or nearly horizontal as in rose comb leghorns. The pea comb is a medium sized, smooth and soft comb having three ridges. The middle ridge is a bit higher, running from the beak to the top of the head, which are either undulated or marked with small rounded serrations. The single comb is the most widely recognized type of chicken comb and is rigidly upright. It is attached to the chicken's skull from the beginning of the beak to the back of the head and features a series of 5-6 fleshy ridges called points and a thicker portion called the blade in the back, often more typical of hens. The walnut comb gets its name from the characteristic furrowing in its surface and is similar to the outer appearance of a walnut shell. It is a medium sized solid comb genetically derived from two dominant alleles for the pea and rose combs typical of silky chickens. The development of comb is controlled by two independently assorting non-allelic gene pairs capital R and capital P on two different chromosomes. The gene capital R gives rise to rose comb and capital P produces pea comb. A cross was performed between rose represented by capital R, small p pattern bearing fowls and single represented by small r, capital P pattern bearing fowls. The capital R and capital B gene for rose and pea combs together produce a new phenotype walnut represented by capital R, capital P in the F1. In the heterozygous state, both the pea comb gene and the rose comb gene can express themselves. That is, only one copy of the rose comb gene or the pea comb gene is sufficient for that type of comb to occur. Therefore, both genes in this case act as dominant genes. When the F1 hybrids are interbred, an interesting result was obtained. The F2 showed walnut, rose, pea and single comb in the ratio of 9:3:3:1. In this, the F2 ratio is not altered, but it is the clear indication of involvement of more than one pair of genes. In F2, the pCOMP gene is absent and one copy of the ROS-COMP gene is present. It results in ROS-COMP. In other words, a ROS-COMP has two possible gene combinations: capital R, small p, or capital R, small p. When the ROS-COMP gene is absent and one copy of the pCOMP gene is present, the result is a pCOMP and has one of two possible gene combinations: small r, capital P, or small r, capital P. One copy of each gene is present in chicken. It results a walnut comp and has one of four possible gene combinations: capital R, capital P, small p, capital R, capital P. Third possibility is capital R, small r, capital P, or capital R, capital P, small p. When both genes, that is capital R and capital P genes, are absent, it results in a single comp and has only possible gene combination small r, small p. In this experiment, the single comp acts as recessive to both capital R gene and capital P gene, indicating that the single comp have recessive alleles for ROS and P. The recessive alleles for both ROS, P in single comp indicates that more than one pair of alleles controlling comp shape. In F1, the production of walnut comp clearly indicates the interaction of two dominant genes, capital R and capital B, for ROS and P. In F2, ROS-COMP pattern is produced by capital R gene together with small p gene, and the pCOMP is controlled by capital P gene with a recessive small r gene. Single comp is produced by the genes capital R and capital P in their recessive state. In this experiment, the inheritance pattern of genes does not differ from Mendelian pattern. The main difference is new character appear in the form of single.